Thank you, Len, for the introduction. And so I would like to spend the next 30 minutes to tell you a little bit about single-cell genomics, and in particular the projects that have been and are ongoing at the JGI. So the first proof of principle study that sequencing from a single microbial cell is possible was in 2005. So that was about 5 years ago. Seems to be a long time, but single-cell genomics of microbial cells is still being considered in its infancy. So it's still a fairly young field. There haven't been very many studies, and that has been in part due to some of the technical challenges of the approach, many of which have been solved or are being solved at the moment. And so I believe right now is a really good time to push forward with this field and this method. So the outline of my presentation today will be, I'll give you a little bit of a background on why we do single-cell genomics and how do we do single-cell genomics, and what have we achieved with this approach as of to date. I then want to talk about the projects that are and have been ongoing at the JGI. In particular, focus on the GEBA uncultured project. That's an initiative that we just recently started. And I'll close off with a kind of summary and future outlook slide. So coming to the background. Why do we do single-cell genomics? I think I don't have to convince most of you why we do it, but I'll give you this brief background anyway. To date, there are we're approaching almost 2,000 genomes that have been sequenced, the vast majority of which are from cultured representatives. We can easily grow them on plates and cultures, recover hundreds of micrograms of high molecular weight DNA for shotgun sequencing. The vast majority of microbes, however, are uncultured as of to date with current culturing methods. So that has been a real bottleneck in sequencing these microbes. So what do we do if we cannot culture them? The approach that has been used for the last 10 years is the metagenomic approach, and that's been extremely successfully used in many, many studies. You just take the environmental DNA and you shotgun sequence it. This is a very powerful approach for gene discovery. You can recover complete genomes depending on the abundance profile of your community and how deep you sequence. And as we also learned yesterday in Eddie's talks, that now with the second generation sequencing platforms and the extreme high throughput, we may be able to soon recover a higher amount of microbial genomes, even from high diversity environments. Now, if you're interested in a particular community member, what people have been doing is target cell enrichment. You can use density gradients, filtration, you can use fluorescent activated cell sorting, and various other methods to enrich a particular member. This methodology does not necessarily work for all community members, so it has its limitation. Now, the idea behind the single-cell approach is that you specifically pick out your target cell from the complex environment, and you amplify its genome and you sequence its genome. Now, the big advantage over this target cell enrichment approach is that if you have a population that you enrich and you have a high degree of heterogeneity within this population, you end up with a composite assembly and a composite genome. While using the single-cell approach, you end up with a true single-cell genome of an individual cell. So now, how do we do single-cell genomics? Very briefly, you isolate your single cell, and there's a range of different methods. I just want to point out at the JGI, we have a micromanipulator that we use for low diversity samples. We also have a fluorescent activated cell sorter, FACS, that we use for higher diversity samples, and we also collaborate with a Bigelow lab that has a single cell sorting facility. Other methods are microfluidic chips, serial dilutions. You can use optical tweezers. More recently, people have been using LCM, laser capture microdissection. Now, once you have your single cell, you lyse it and you amplify it. Whole genome amplification method that we use is the MDA, multiple displacement amplification. This is an isothermal amplification. We run these MDAs at 30 degrees Celsius using a strand displacement polymerase, Phe29. So all you need is your template, your DNTPs, your primers, and we use random hexamer primers, the enzyme, and the whole reaction takes off, and you end up with micrograms of DNA that are derived from only a single cell, so only a few femtogram of DNA. So sufficient material for shotgun sequencing. Now, I like to put up this slide. It's my key challenge slide, just to point out the approach may sound easy, but there have been various challenges to the approach at various stages, so various flavors of challenges. Contamination is a big problem. You can imagine if you have only one copy of your genome, that any little fragment of DNA that's derived from wherever will start competing with your amplification. The lysis is a limiting factor. Right now there is no, or we don't have a universal lysis method that works for all taxa. So with the method that we use, we cannot capture all the taxa that we would like to. The MDA itself has a few disadvantages. It introduces chimeric rearrangements. Those are largely inversion deletion events. And we have also experienced contamination in the reagents, which has been a challenge. And there's a bias. The MDA does introduce a random bias, especially on the single cell level. So you get parts of the genome that are highly overrepresented and other parts that are highly underrepresented. But as I mentioned, a lot of these challenges have been addressed and are being addressed in many labs around the world. What we, for example, do at the JGI, we have a decontamination protocol for the MDA bias. We do normalization, as was done at MIT. So there are a lot of workarounds to make this approach a little bit more solid. So just to give you an overview of what have we done with the approach to date. As I mentioned, the proof of principle study that you can do this from microbial cells was in 2005 in the Alaskan lab. And there have been a range of very nice studies that the first ones were resequencing, for example, Prochlorococcus and E. coli, TM7, and then a flavobacterial study, which I'll talk a little bit later, and a Prochlorococcus study. What I want to point out on this slide is what did we recover in all these different studies with respect to how much of the genome did we get, and how well did it assemble? Researchers or scientists are able to recover up to 95% of the genome, but it's fairly fragmented. So we generally end up with hundreds and thousands of contacts, and that is in part due to the MDA bias. You don't get as good of an assembly as you would get with an isolate genome or DNA from an isolate genome. So what are the projects that have been ongoing at the JGI? I want to introduce you to 3 studies. One that involves two marine flavobacteria, one that involves the sharpshooter symbiont, and then a project that we, as I mentioned, recently initiated, the GEBA uncultured project. So the marine flavobacteria. In this study, we worked with Ramona Stepanauskas from the Bigelow laboratory, and they collected marine flavobacteria from Booth Bay Harbor in a 1 meter depth, and took these cells and sorted them. Here they did a Cyto13 stain and sorted this HNA, the high nucleic acid population, to recover single cells. These were amplified and shotgun sequenced at the JGI. At the time, we did a Sanger 454 hybrid approach, and we were able to recover 2 draft genomes that represented about 80, respectively 90% of their genomes. And the assembly was fairly contiguous. We recovered contigs that were in the range of 0.6 megabases, so considering it's from a single cell, we thought that was very good. But I'd like to point out that this assembly didn't come together by itself. It required a significant amount of finishing efforts. What you see here on the left is the assembly as it was before finishing efforts, so it was fairly fragmented. You see a lot of contigs. This assembly went through some finishing efforts at our partner lab at Los Alamos National Lab. There were 5 rounds of adaptive PCR. There were various shadow libraries that were generated to close a lot of these gaps and bring these contexts together. So we did recover 2 high-quality draft genomes, but it did require a significant amount of finishing efforts. Now, after we finished this project, we had Nancy Moran and Howard Ochman at the JGI. They were on sabbatical, and Nancy has been working with these sharpshooter symbionts for quite some time, and she's very interested in the system. And we were looking for some samples and collaborations, and we thought, well, this seems to be an interesting system. They have symbionts that have fairly small genomes, and maybe these may be good targets for the single-cell approach. So we started working with her and also Howard Ochman on these green sharpshooter symbionts. So the green sharpshooters, they are a very prominent group of sap-feeding leafhoppers, and sap is a very nutrient-poor nutrition. So how can they live off sap? Well, they associated themselves with these two symbionts. One is a flavobacterial symbiont called Solcia milleri, and the second symbiont is a proteobacterial Baumannii. And these symbionts form together with the host this nice tripartite symbiotic relationship where the symbionts provide the host with various nutrients. So it's been described in a related host system, the glassy-winged sharpshooter, that the Solcia provides the amino acids for the host, that it's lacking in its nutrition, and the Baumannii provides cofactors and vitamins. So we were studying the green sharpshooter symbiont. Again, it harbors these two symbionts. It is a vector for Xylella, which can cause very serious diseases in plants. In California, for example, it causes Pierce disease, which has been a real threat for the wine industry since it infects the grape wine. And it has been a real threat, and it's in particular for Chardonnay, I believe. So we thought, well, the Solcia symbiont may be an ideal candidate to test the single-cell genomics. And in particular, we were wondering, well, can you recover a complete genome from a single cell? So Solcia is ideal. It's uncultured. It has a very small genome size. The single-cell isolation is quite simple. We can easily identify it under the microscope, and using the micromanipulator, we can isolate single cells. We have fresh sample available. These leafhoppers are kept at UC Berkeley, so we have easy access to fresh sample. And these genomes are polyploid, so they have many copies of their genome as we tested using digital PCR. And what you see here, this is just the glass capillary of our micromanipulation system that allows us to pick up individual cells. So what did we do? We took one sharpshooter, we dissected the bacterium that harbors the symbionts, and we pulled out a single cell, a single Solcia cell, amplified it, and sequenced it. Now in parallel, we did the metagenomic approach. We took 25 sharpshooters, dissected the bacteriums, pulled them, extracted the metagenomic DNA, and did shotgun sequencing. And then we were wondering, well, so are these two genomes identical that we derive using metagenomics and single-cell genomics? The single-cell genomic approach, as I mentioned, the MDA introduces chimeric rearrangements. So we wanted to make sure that the single-cell genome that we recover is not an artifact of some of the MDA-derived chimeric rearrangements. So we compared the two genomes, and I'll talk about this in a little more detail, but I want to point out we did recover a complete genome, a closed genome, for using both approaches. And we found 4 different sequence variations. 2 of them were encoding sequences, 2 of them were non-coding sequences. And these variations are essentially the copy number changes, their copy number variations in small repeats. And so we concluded that these are true, genuine biological differences and not artifacts that are derived through the MDA. These samples were collected at different time points, so that could explain this slight variation. And one thing I would like to point out before I move on is that, again, for the single-cell approach, we required one symbiont from which we isolated one cell, while to do the metagenomic sequencing approach, we had to pull 25 animals to recover enough metagenomic DNA for shotgun sequencing. So as I mentioned, we did recover a complete genome. It didn't come together by itself. It required a little bit of finishing efforts. So we sequenced the single-cell genome using a Sanger and 454 hybrid approach. We assembled it. It ended up in 31 contigs, so we still had gaps. And then we used Sanger and Illumina to close the gaps and also to polish the genome. And what you see on the right is the genome, and these outer rings recover the sequence coverage, the Sanger 454 and Illumina sequence coverage. As you see, the amplification is very biased, so parts of the genome are highly overamplified, while other parts of the genome were less amplified. This region in particular had very low sequence coverage and was the region with the majority of the gaps. This inner ring of orange and green tick marks represent the captured and the uncaptured gaps in this genome. So now we have a complete finished single-cell genome that we pulled out of a population, and we have metagenomic reads. Now we were wondering, is there any variation, any sequence variation? So we took the single-cell genome as our complete reference. We took the metagenomic reads and mapped them back to the single-cell genome to look for variation. We only found 2 SNPs within the whole genome, and that indicated that within this spatially very defined social population, there is a very low genetic diversity. But you could imagine doing this with a population that has where you have higher heterogeneity, and it could be very interesting to use your single-cell genome as your reference and then look at variation by mapping back the metagenomic reads. Now I want to switch gears and talk about the GEBA Uncultured project. Most of you are probably familiar with the GEBA project. It's the Genomic Encyclopedia of Bacteria. This is a project that was initiated by Jonathan Eisen and his lab, as well as Phil Jugenholz and Nikos at the JGI. And the idea or the goal is to sequence 1,000 bacterial archaeal genomes that are from very diverse branches of the tree of life, so really expand sequence space phylogenetically. There was a pilot of 56 genomes. Those were 53 bacterial and 3 archaeal genomes that was recently published. Now, why do we need GEBA? Why do we need to sequence all these genomes that are phylogenetically diverse? If you look at the current phylogenetic tree of life of the bacteria and archaea, and each one of these lines represent phyla. In blue are the phyla that are fairly minimally sequenced or have very few sequenced representatives, if any, while in red are the very well-sequenced phyla. And this is the tree that shows the archaeal and bacterial phyla of all the cultured isolates. So it becomes apparent that the vast majority of the sequenced genomes to date are only from 3 different phyla, from the actinobacteria, the proteobacteria, and the firmicutes. So this really explains why we need to expand sequence space to have better reference genomes that are phylogenetically diverse. And what you see here now, I hope you can see that the colors changed. This is the tree as it looks post-GEBA. So now a lot of these phyla are covered in red, which means we aim to populate them with reference genome data sets. So the question is, what is the value of that? Why should we do that? First of all, having these reference genomes, and for most of you who work with a lot of metagenomic data, you will probably appreciate having these reference genomes, as they will serve as phylogenetic anchors for metagenomic data. They will help us improve annotation and give us an improved understanding of evolutionary diversification, so the origin and evolution of genes, lateral gene transfer, and so on. And they will also accelerate the discovery rate of novel genes, proteins, protein families, and pathways. And what you see in this plot here at the bottom right is the discovery rate of protein families. So on the y-axis, you have your protein family discovery rate. And that is being plotted for a number of genomes and as function of the phylogenetic breadth. So this first plot you see here is on the species level. Then you have it plotted for the family level, for the phylum level, in this case the actinobacteria, and lastly for the domain, so taking all the GEBA genomes. And what's clearly apparent is that the more phylogenetic breadth you have, the more accelerated is your discovery rate. Now, one question that remains is how much diversity do we still need to sample? What you see here is the phylogenetic diversity for 4 different populations. So first of all, in blue, The phylogenetic diversity is plotted for the pre-GEBA genomes, so all the microbial genomes that have been plotted before we started sequencing all the GEBA genomes. And you see that it levels off fairly quickly by resequencing various strains. Then in red, the curve becomes much steeper, and these are our GEBA genomes. So suddenly we're increasing phylogenetic diversity. And then in dark gray, you see all the organisms for which we have 16S sequences for, from all the cultured organisms. So the curve becomes significantly steeper, and even more so once you add all the 16S sequences, so all the organisms that we have currently in the database. So now if we look at how much, again, if we ask this question, how much diversity do we still need to sample? And again, these numbers are constantly changing. This is as of to date, but this is a constantly changing model. So to capture about 50% of the culture diversity, we'd have to sequence in the range of 1,500 genomes right here. If we would want to capture 50% of the complete, as we know to date, the whole diversity, including the uncultured microbes, we would have to sequence about 9,000 genomes. Now, 1,500 genomes may be a tractable goal over the next many years. 9,000 genomes, that will probably take us a while, but it could be a goal. So now the next question is, how do we even capture this diversity? And that brings me back to our single-cell approach. So what you see here is a phylogenetic tree that captures not only the phyla of the cultured representatives, but of all bacterial and archaeal organisms. And you can see that there are many trees that we haven't, we don't have any cultured representatives, we don't have any sequenced representatives. And if we want to extend GEBA beyond the cultured microbes, we need to use an approach like the single-cell approach to be able to set these reference genomes. So this calls for the GEBA Uncultured project. Here the goal is to sequence about 500 bacterial and archaeal genomes from phyla without any cultured representatives. A pilot is 100 genomes, and the approach will be the single-cell approach to ultimately fill in the tree of life. Where are we with this project, and what is the plan? We have selected 6 different environments, and behind each one of these environments is a collaborator that we're working closely with on getting fresh sample. Steve Hallam, I don't want to go through the list, but you can have a look at it. We have samples from Yellowstone, from Dussel, which is the Deep Underground Science and Engineering Laboratory, Great Basin, Saginaw Lake, and so on. So each one of these environments have been sampled. Some different samples are in various stages, but the idea is that we extract metagenomic DNA to do pyrotag. And for a lot of these environments, we already have preliminary pyrotag data that tells us that these environments are highly enriched for candidate phyla representatives. And then we also sample the cells for single-cell sorting, and these are anonymous sorts, so we just sort thousands of cells that we then screen, and we look for representatives that are good candidates for the GEBA Uncultured project for shotgun sequencing. And just to give you some very brief preliminary data, in this plot you see the percentage of candidate division representatives. This is based on pyrotag data of 2 of these environments. And you can see that these are heavily enriched for unknown uncultured microbes. For example, in the case of the Saginaw Lake, we have a lot of the OP representatives, OP 8, 9, 10, and OP3. And so these are the ones we are after. And what is the status with respect to sequencing? We already, I just want to briefly point out, we already have 13 of these single cells that are in the queue that are from various different candidate phyla. And to close off, where do we want to go in the future? We'd like to improve the methods, including the throughput at the JGI. We want to use the single cell. We will be using single-cell genomics as the enabling technology to populate the tree of life with additional reference data sets from uncultured microbes. We see the single-cell approach as a very powerful complementary approach to the metagenomics approach, and we would like to use both at the JGI in parallel. And you can imagine that disassembling a metagenome into its single-cell components can be extremely powerful. And we'd like to pursue newer technologies, such as targeted single-cell genomics and single-cell transcriptomics. And just to show one more slide, for those of you who have already looked at the new CSP call, we are this year, it's maybe hard to read, but we're requesting ideas, projects, proposals that involve single-cell sequencing. So we're very interested in pushing this approach forward and working with collaborators on these projects. And I'd like to acknowledge a lot of people at the JGI, at Los Alamos, University of Arizona, UC Berkeley, at the Bigelow Laboratory, and also the GEBA uncultured community and the Department of Energy. And I'll take questions.
Do we have any questions? So one note while people are putting their hands up, there was a BlackBerry potentially lost in or around this room, so if anybody found one, please bring it to me and I'll take it back to its owner.
Hi. I'm very interested in single-cell sequencing in sort of a high throughput way as maybe a complement to metagenomic sequencing or other community sequencing efforts. And I wonder what you think about methods like, I don't know if you've explored cell sorting or other sort of high throughput ways to isolate individual cells and then enter them in a pipeline for genome sequencing so that they're sequenced in parallel and you can assess things like diversity within a population of single cells at the genomic level.
So I mean, the most high throughput way I envision right now is, would be to do your high throughput sort in 384-well plates, lyse it, amplify. I mean, you could think about microfluidic chips even, but you need to get them off the chip to get them on the sequencing machine. You could do barcoded Illumina libraries that you pool for shotgun sequencing, but there is a transition where you need to get it either off the plate or off the microfluidic chip to get it into the sequencing queue.
Hi, I was curious about when the sharpshooter project, when you were trying to do both the metagenome and the single cell, was the metagenome helpful in trying, or could it be helpful in trying to do the assembly and the finishing, or do you think the same biases or artifacts or realities of gap formation propagate?
I think it would have been helpful. We recovered the single cell genome first, so at the time, we weren't using the metagenome data yet to assist the single-cell assembly, but clearly, since the biases are most likely different in the two approaches, that you would use the metagenome to assist. You could assist the single-cell assembly and finishing, absolutely.